In this video, I will briefly discuss proteins. So the monomer of a protein is an amino acid. This is the building block to make our proteins. An amino acid starts with a central carbon that you can see there in black. Attached to that central carbon are 4 different groups. In red, you can see the carboxyl group. In blue, you can see the amino group. In green, you can see the R group, which is the side chain that varies between every amino acid and gives that amino acid its specific properties. And that central carbon is then also attached to a hydrogen ion. So this makes up our amino acid. Well, there are 20 specific amino acids in the human body that are important to the function of proteins. When we put these amino acids together, if it is a short chain, we form a peptide. If it is a long chain of amino acids, we call this a protein. Our amino acids are held together almost like beads on a string by peptide bonds. So this sequence of these amino acids is the first step of our protein structure. We call this our primary protein structure, and it is just the amino acids that are in order held together by peptide bonds. These proteins can be very large, consisting of hundreds and thousands of amino acids. So we have to fold them so they can fit into the place they need to fit and do their job. So our secondary structure of proteins involves folding into 2 main types. The sequence of amino acids will be folded into an alpha helix or a beta pleated sheet form. From there, we will go to our tertiary or third step of protein structure, and this is where our protein will be folded and given its 3D shape. Proteins need a specific shape to do their job. Structure leads to function with everything in the body, starting at the level of molecules. So the proteins are built into their specific shape to do their specific job. There's an optional quaternary or 4th level of protein structure where we take multiple tertiary structures and add them together to make a large protein unit. Again, proteins have to be a specific structure to perform their specific function. Their structure is key to their function. So if a protein is denatured or it loses its structure, it then also loses its function and is unable to do its job in the human body. An example of things that would cause protein to be denatured are too high of a temperature or too high or low of a pH. Proteins are very specific in the conditions that they work, and any alteration to that can cause them to become denatured and lose their structure and then their function.